Every day in our microscopic world, a single bacterial cell can grow and multiply to form millions of itself over time. This is called binary fission. As the bacteria divide, so will the chromosome, giving each daughter cells a copy of the parent DNA. This is called DNA replication. What if you wish to artificially replicate a single strand of DNA to produce multiple copies of itself? In 1985, Carey Mullis invented polymerase chain reaction. The invention of PCR has paved the way for diagnostic application, especially in our recent fight against COVID-19 and genetic fingerprinting in forensics. Assuming you wish to amplify the DNA of an unknown bacteria, you first need to extract and purify the DNA from the organism. The first step is cell lysis. Lysis is the disruption in the cell membrane, often done by enzymes or detergents. Proteinase K and RNase A is often added in the lysis step. Proteinase K is added to digest contaminating proteins in the cell lysate, while RNase A is added to degrade RNA because when RNA is eluted together with DNA, it overestimates the DNA yield in downstream processes. When you add lysis buffer into your bacterial cell and incubate, this process frees up the intracellular components such as proteins and DNA. This end product is called cell lysate. DNA binding buffer containing cheotropic salts will be added to the cell lysate. The mixture is transferred to a column, the sample is then centrifuged. In this instance, the column containing silica membrane, the nucleic acids will bind to the silica membrane in the presence of high cheotropic salts, which provides a high salt concentration environment. Although the surface of the silica membrane and the DNA is both negatively charged, but the cheotropic salts will disrupt the hydration shell around all the molecules, forming a cation bridge between them. The flow-through will then be discarded. A washing step containing alcohol is performed to remove proteins and other contaminants that could be trapped in the silica membrane. Centrifuge then discard the flow-through. The wash step is then repeated. Transfer the column to a new collection tube. Elution buffer is then added. Having a lower elution volume will give you a higher DNA concentration. Incubate for 1 minute, then centrifuge for another minute. High pH and low ionic strength buffers are often used to elute DNA as the silica surface will have a stronger negative charge at higher pH due to higher levels of hydroxide groups, resulting in an increase in repulsion between the DNA and silica surface. We will now have the eluted DNA that is pure enough for the use in PCR. There are 7 key components often found in a conventional PCR master mix. First, we have PCR buffer. It provides an optimal chemical environment for DNA polymerase to function. Buffer pH is between 8 to 9. Sometimes, chemical additives are included in the buffer to improve amplification specificity. Examples of PCR enhancers are ammonium sulfate and dimethyl sulfoxide. The composition of the buffer varies with the type of polymerase used. Different companies produce different types of PCR buffers. It is therefore important to follow the recommended guidelines from the company. Second, we have DNTPs. DNTPs are deoxynucleotide triphosphates. DNTPs consist of DATP, DTTP, DCTP, and DGTP. Adenosine, thymine, cytosine, and guanine are bases of DNA. DNTPs are important during the extension phase when bases are inserted into the backbone of the DNA. Third, we have primers. Primers are short sequences of nucleotides, generally about 20 nucleotides long. There are 2 types of primers in a conventional PCR reaction, the forward and the reverse primer. When designing a forward primer, the sequence of the forward primer is the exact copy of the 5' to 3' DNA template. But when designing a reverse primer, the sequence of the reverse primer must be reverse complemented of the 3' to 5' DNA template. Fourth, we have DNA polymerase. A commonly used DNA polymerase is Taq polymerase. The structure of Taq polymerase is often described as a hand. The fingers and thumb make up the polymerization domain. The 5' to 3' exonuclease domain digests nucleotides in the 5' to 3' direction ahead of polymerization to remove 5' flaps that are overhanging. The 3' to 5' exonuclease domain digest nucleotides in the 3' to 5' direction, which acts as a proofreading function to check whether if the correct nucleotide has been inserted into the newly synthesized strand. Fifth, we have magnesium chloride. Even if magnesium chloride is present in the buffer, additional magnesium chloride is added to optimize polymerase activity. A pair of magnesium ions acts as a cofactor for the DNA polymerase by enabling incorporation of DNTPs during polymerization. Lastly, we have our purified DNA template, which is often added last. Thermocycler is essential in the field of molecular biology. It is better known for the use in amplification of DNA, hence it is often referred to as a PCR machine. In the early days, PCR was performed using water baths of varying temperature. Now, PCR machine has thermal block that allows for a greater and finer temperature control. The lid on the machine will be heated to prevent condensation from forming on the inner side of the tube lid. When preparation of the master mix is completed, the PCR tube is placed into the thermal block. This graph shows a typical PCR thermocycling profile. The first stage is initialization at high temperature. This step is important to activate hot start DNA polymerase. In the presence of high heat, only then enzyme activity will begin. Using hot start DNA polymerase prevents primer-dimer formation at room temperature. Primer dimers are formed when complementary bases anneal to each other and polymerization occur, affecting the yield as lesser primers and polymerase are available during the reaction. The second stage is denaturation. It occurs at high temperature to break the hydrogen bonds holding the two strands together. This forms two single-stranded DNA. The third stage is the annealing of primers. It occurs at a temperature depending on the melting temperature of the primers used. It can be determined using a calculator online or perform a temperature gradient test to determine the optimal annealing temperature. In the annealing stage, forward and reverse primers will bind to its complementary sequence on the DNA strand. The fourth stage is extension. Extension temperature is often at 68 degrees Celsius to reduce DNA damage. In the extension stage, DNA polymerase incorporates DNTPs in the 5' to 3' direction to form the complementary strand. The last stage is final extension. The final extension allows for better yield, especially for longer DNA strand. Observing DNA strand with our naked eye will be impossible. With agarose gel electrophoresis, it allows for the separation and visualization of DNA amplicons based on size. To prepare the agarose gel, agarose powder is mixed with TAE buffer. In this example, 1 gram of agarose powder and 100 milliliters of 1 times TAE buffer forms a 1% agarose gel mixture. Different agarose percentage is used depending on the size of the DNA. For example, 2% agarose gel is used for smaller DNA size of 100 base pairs. Higher agarose percentage allows for better separation of smaller DNA. The mixture is then heated using a microwave. The agarose liquid will be poured into a casting tray with a suitable well comb. The number of wells is dependent on the number of samples. In this step, fluorescent nucleic acid stain is mixed into the agarose liquid. Fluorescent nucleic acid stains are safer alternatives to the toxic ethidium bromide often used in the past. The agarose gel will solidify after 30 minutes. Once the gel has solidified, it will be placed into an electrophoresis tank. The setup requires a power supply that is connected to a cathode, the negative end, and an anode, the positive end. Once the agarose gel is prepared, the DNA is loaded into the wells of the agarose gel together with loading dye. DNA loading dye often contains glycerol, bromophenol blue, and xylene cyanol FF. Glycerol in the loading dye increases the density of the sample, ensuring that minimal samples are lost during the loading step. Both bromophenol blue and xylene cyanol FF acts as a tracking dye to allow the user to visually ensure the DNA sample does not run off the gel. When an electric field is applied, the negatively charged DNA will migrate to the anode, the positive end of the tank. The smaller the DNA fragment size, the further it will migrate down the gel. Larger DNA size will remain at the upper region of the gel. In order to visualize the results of the gel, we will use a gel imaging system that emits UV light. allowing you to see the fluorescent DNA bands. This is an example of a typical agarose gel electrophoresis results. The DNA ladder at the left will allow you to determine the size of your sample based on predetermined marker size. The sample in this example is shown to be around the 750 base pair marker.